So today we're going to continue our unit on genetic engineering. We talked about the other day, genetic engineering is designing living organisms to fit the needs of humans. And we talked about selective breeding, how humans have used kind of this method of selectively breeding certain organisms together to get desired traits out of organisms. Whether we do that with different breeds of dogs to get certain traits, like a fast dog, a slow dog, short dogs, or we do it with things like maybe cattle to get cows that produce a lot of milk or produce a lot of meat, or different examples of that. Corn was an example, pigs are an example. Today we're going to talk about a different method of genetic engineering, something called recombinant DNA. And so we're going to talk about what this is, and it's a little bit out there. It's going to sound a little crazy, but I swear it's a real thing, and it can really be done. And we'll actually, there's going to be 2 videos on recombinant DNA, and this is going to be just part 1 of those 2 videos. So let's talk about recombinant DNA. What is genetic recombination? What is this concept of genetic engineering through recombinant DNA? So we talked about selective breeding, using that to kind of manipulate organisms. But one limitation of selective breeding is that it only allows us to take traits that already exist in a species. We can magnify those traits, we can hybridize certain organisms to mix traits, but we're limited to what traits exist inside that population. Now, if there's a mutation, that means that organism gets a new trait, and then we can breed that through. But what happens if we want a trait that doesn't already exist in the species that we're looking at? So what if there's a trait that we want maybe inside our pet dog, but doesn't exist in dogs? To do that, we use something called recombinant DNA. And recombinant is just another word to mean to recombine. Recombinant DNA is when we join together DNA from 2 or more sources. And that's what makes it possible to change the genetic composition of living organisms. What do I mean by joined together from 2 or more sources? I mean literally from 2 different organisms. So we can take the DNA from 2 different organisms and combine them together to make a mix of the 2. I'll give you a very crazy example of this, but it's absolutely a true example. Some scientists took the DNA from humans that codes for the human ear, and they inserted that DNA into the DNA of a mouse. And they produced that mouse baby, and that mouse grew a human ear on its back. And this is a real, true example. This is a real mouse. This isn't a fake picture. This has really been done. So they were able to combine the DNA of humans with the DNA in the mouse. Now, why do this? Why grow a mouse on why grow an ear on a mouse? Actually, it was a real-life example of scientists were trying to be able to replace the human ear in case anyone lost it. If your ear got cut off, they could grow you a new one on the back of a mouse and then attach it to the human. So this is a pretty crazy example, but it's an absolutely true example of recombinant DNA technology. And there's actually many more examples of this that you can go see that are really crazy. You'll see a couple today. Glow-in-the-dark cats, glow-in-the-dark plants, where they use the DNA from a glowing jellyfish to make all these organisms glow. So there's plenty of examples out there. There are strawberries that have the DNA of fish in them so they don't freeze in the winter. So crazy examples of this happening, but it's a real thing in science you could actually do. So let's talk about how this is actually done. How do you combine the DNA of 2 organisms together? And it's actually a 3-step process. So we're going to talk about these 3 steps in detail, but the steps in general to make a recombinant DNA is the first step is that you need to remove the DNA from both organisms. So you need to remove the DNA from their cells. You need to remove the DNA or the gene from the donor, the species that you have the desired trait, and you need to remove the DNA from the host. So let's say if we were talking about the mouse in the ear, we need human DNA for the ear, and we need to cut out the mouse DNA as well, or remove the mouse DNA. The second step is to cut and paste that DNA together, and we use something called a restriction enzyme to cut and paste the DNA together. And then the third step is to put the DNA back into the host organism, put the DNA back into a host cell, usually an embryo that can grow into a new organism. So we're actually talking about just steps 1 and 2 in this video. We'll talk about step 3 in another video. So let's focus on step 1, which is removing the DNA from the cells. So before we can start cutting and pasting DNA together, we need to remove the DNA from both living organisms. So removing the DNA from both the donor and host cells is actually quite simple. Scientists go through a process called DNA extraction, which we may or may not have talked about in a previous class, but the process is pretty simple. Just recall that inside your cell here you have the nucleus, and the DNA is inside that nucleus. So when scientists are trying to extract DNA, they have to break into the cell, into the nucleus, and get the DNA out. It turns out to be a 4-step process. The first step is only if there's plant cells, because remember that plant cells have an extra layer of protection called the cell wall. So in order for scientists to get the DNA out, they have to break through that cell wall. And they can do this actually by just physically smashing the cell, and that'll break apart that cell wall. And the second step is to get through the membrane of the cell, to get that through that outer layer of protection. We talked a lot about how the membrane is made of lipids. It's made of these lipid molecules that allow certain things out and certain things in. Turns out that those lipid molecules are actually very similar to these kind of fat molecules and grease molecules, and so using something like a soap or a detergent actually removes those grease molecules away, or those lipid molecules away. The same way you kind of like wash your dishes with soap, and it gets the grease off all your dishes. If you have a really greasy pan, you wash it with soap, and it just carries it all away. The same thing happens there. And then the third thing is that you need to break up all the proteins that are holding the DNA in place. So remember, we talked about how DNA is supercoiled, and it's supercoiled around these proteins called histones. So they use an enzyme called protease. Sometimes it naturally occurs in things like pineapple, and you can actually use that to break apart the proteins. And then it's just a simple thing of getting the DNA out from both cells. You can usually just extract it. They extract it using something called a centrifuge, which spins a test tube around really fast so the DNA all sinks to the bottom, and then they can extract it out. So it's a pretty simple process. So once you've extracted the DNA from both the host cell and the donor cell, now it's time to cut and paste that DNA together. So to do that, we're going to cut the DNA using something called a restriction enzyme, and we're going to talk about how that's possible. So cutting and pasting DNA together, and this is kind of how, we have the DNA from maybe our mouse and our DNA from our human. How do we combine the two together? How do we cut pieces out of the human DNA and insert them into the mouse DNA? So to do that, I said we're going to use something called a restriction enzyme. And restriction enzymes are chemicals that are capable of cutting up DNA. And restriction enzymes, you're going to find out they're naturally occurring. They exist in bacteria. So you're going to see the restriction enzymes have all these weird names. They're named after the bacteria that they're found in. And one thing that I just want to go over before we kind of talk about how they work is, why might bacteria have an enzyme to cut up DNA? Where do these restriction enzymes come from? Well, it turns out that one of bacteria's biggest enemies are viruses. And the way that viruses infect cells is by injecting their DNA into the cell. And they trick the cell into reading the virus's DNA and making more viruses. So as kind of a way to fight off viruses, bacteria have these things called restriction enzymes that will cut up little pieces of DNA that don't match their own DNA. So if a virus tries to infect a bacteria, that restriction enzyme might recognize that virus's DNA and cut it up into little pieces so it can't be used. So these restriction enzymes are naturally occurring in bacteria. You can see here's the virus's DNA. A little restriction enzyme a chemical that cuts up DNA, and it cuts it up in these specific places so it doesn't actually cut up its own DNA. So restriction enzymes are useful to us because they only cut DNA in very specific locations. Like I said, it's a way for them to cut the virus's DNA. They cut at a very specific set of bases. So restriction enzymes actually look for DNA sequences and only cut in those spots. So for example, this sequence here, GGCC, might be a DNA sequence that a restriction enzyme cuts at. And it cuts, kind of right through and cuts the DNA in half. You can see here, HAE3, so restriction enzyme that cuts at the location GGCC. And what's important to note is that I'll use this notation 5 to 3. Remember that DNA strands run in opposite directions. So GGCC, and then if you read it on the bottom strand, you read right to left, GGCC. And one of the interesting things about these restriction enzymes is wherever they cut, it's always a palindrome. So if you read it one way and read it the other way, it's the exact same thing. GGCC, GGCC. It'll always look like that, which is just kind of something interesting. So this enzyme cuts at GGCC, and it cuts it right in half. And we're going to talk about this, but whenever DNA gets cut right in half, right down the middle, we call it a blunt end. Now this is going to be important because blunt ends can't be joined back together. So we're actually going to look at an enzyme that cuts a different way, EcoR1. And this makes a staggered cut, so it produces what we call a sticky end. So in this case, this enzyme cuts at the sequence GAATTC. If you read it backwards, GAATTC. But it actually cuts not in the middle, but only 2 bases in. It cuts between the G and the A. So it cuts between the G and the A on this strand, and on the bottom strand it cuts here. And this makes a zigzag cut. We call this a sticky end. You can see the 2 examples here. A blunt end is when the DNA gets cut straight through. A sticky end is when the DNA gets cut in a zigzag shape. And this is the one that's important because a sticky end can stick back together. So they're the ones that are useful for genetic engineering or genetic recombination because those sticky ends will paste back together. They kind of fit like puzzle pieces. You can see that's been cut here. Those T's will pair up again with those A's, those T's with those A's again. These 2 pieces of DNA can stick back together like glue. So the rule is this, that if any 2 separate pieces of DNA are cut using the same restriction enzyme and they have the same sticky ends, those 2 pieces will stick together and become one DNA molecule. So you can see here if maybe I had my mouse DNA in blue and my human DNA in red. If I cut those 2 molecules of DNA with the same restriction enzyme and they have the same sticky ends, they can stick together and become one new piece of DNA. So that's the second step of cutting and pasting the DNA together using restriction enzymes. And like I said, those are the only 2 we're going to talk about in this video. In the third part of the video, the second part, part 2, we're going to talk about how to put that DNA back into a host organism and grow it into, let's say, a mouse with an ear. But before we end this video, I want to focus on restriction enzymes and show you really quickly how they work so you're able to model it on your own. So we're going to go over how to practice using restriction enzymes or how they're used at least visually on paper. So you can see here, and I might give you a problem very similar to this. I'm going to always give you a restriction enzyme. It's always going to have a funny name named after the bacteria. This is HPA1. And then the other thing I'm going to give you is its sequence that it cuts at. So you can read here that this enzyme cuts from the 5 direction to the 3 direction at the sequence GTTAAC. And this little dash right here means it cuts between the T and the A. So remember that DNA is always read from 5 to 3. So the top strand, 5 is on this side, 3 is on this side. On the bottom strand, 5 is on this side, 3 is on this side. So we're going to read this strand backwards. So watch how we do this. I'm going to go ahead and read from 5 to 3, and I'm going to look for the sequence GTTAAC. Wherever I see that sequence, I know I'm going to make a cut between the T and the A. So let's go ahead and see if we can find it. GTTAAC. I found it right here. GTTAAC. AAC. I cut between the T and the A. GTT, AAC, cut between the T and the A. I'm going to make a cut right here on my DNA. Then I'm going to keep reading because I want to see if I see that sequence again. It may be there more than once, and if you look right here, GTT, AAC. I cut between the T and the A. So this restriction enzyme is going to cut in two places. Now that's my top strand. I also need to do the same to my bottom strand. So let's read the bottom strand and look for GTT, AAC. I see it right here running backwards because remember, 5 to 3, the bottom strand runs backwards. GTT, AAC, cut between the T and the A. So I'm going to make a cut. And you can see this cut lines right up with the cut on the top, or it should if I typed well. So I should expect to see one right over here. GTT, AAC. So there's my sequence cut between the T and the A. And these should line up and be a straight cut. This should be a blunt end because I'm cutting right in the middle of the sequence, GTT, AAC. I might have not lined up the letters perfectly here to see it. So I'm going to always ask you two questions. How many pieces of DNA did we make when we cut using our restriction enzyme? We could count it up. Here's piece number 1. Here's piece number 2. Here's piece number 3. So we made 3 pieces of DNA. And like I said, they are blunt ends because we cut straight through. Let's look at one more example of how to use a restriction enzyme. Here I've given you a different restriction enzyme, PST1, which cuts from 5 to 3 at the sequence CTGCAG, cutting between the A and the G. So again, I'm always reading from 5 to 3, looking for this sequence CTGCAG. CTGCAG. I see it right here, CTGCAG. So I'm going to make my cut now between the A and the G. CTGCAG, right here. And then I'm going to keep reading and see if I see it again, which I don't believe I do. It's just that one time, CTGCAG. So now I'm going to read backwards from 5 to 3, looking for that same sequence, CTGCAG. CTGCAG. So there's my sequence running backwards on the bottom strand from 5 to 3. And again, I'm going to cut between the A and the G. So I'm going to go ahead and make that cut between the A and the G. Now what you should notice is that these cuts are off-center. So this is an example of cutting using a sticky end. And remember, because if you look up here, I'm not cutting in the middle of the sequence. I'm cutting off-center here, 4 letters on this, 5 letters on this side, 1 on this side. I'm going to have a blunt end. So again, 2 questions. How many pieces of DNA, and are they blunt ends or sticky ends? How many pieces of DNA? I have 1 on this side. Here's my second. I have 2 pieces of DNA now. I've cut it in half, and they are sticky ends because I cut in that zigzag shape. So that's just an example of how you go ahead and use these restriction enzymes. Now I'm going to give you a chance to practice using those restriction enzymes, cutting up sequences of DNA given certain restriction enzymes.